Okay. Okay, good afternoon. After the link
Good
of
afternoon, sir.
discussion, it took 25 minutes. Still only 16 have
Good
joined.
afternoon, sir.
That means the remaining 10 students are not bothered to join the class event. Who are they? ఈరోజు ఎవరైతే 10 మంది క్లాస్ జాయిన్ అవ్వలేదో వాళ్ళు ఎక్స్ప్లెనేషన్ ఇవ్వాలి. Okay. So yesterday we have discussed about the structural organization of electron transport chain. Okay. So what are the two important components of electron transport system? Kirtana. What are the two important components of electron transport system?
NADH and FADH2, sir.
It is the respiratory complexes and the mobile carriers. The
Mobile carriers, sir.
first component is respiratory complexes.
Respiratory complexes and mobile carriers, sir.
Mobile carriers. Good. So what are how many respiratory complexes are present? Surya?
Respiratory complex
Hi,
1,
sir.
2.
5. Good. Very, very good. Number of respiratory complexes is 5. Next, what is the name of the first complex? Sujana.
Sir, NADH coenzyme reductase.
NADH coenzyme Q reductase. What are the other components of this complex?
Sir,
Components of complex 1. Other
sir,
components.
voice.
ఇంకా ఏం కాంపోనెంట్స్ ఉన్నాయి కాంప్లెక్స్ 1 లో? ఎనీబడి?
Sir, iron.
Sir, FMN and 6 to 7 iron sulfur clusters, sir.
Very good. Excellent. Next, Chaitanya. What is complex 2?
Succinate COQ, sir.
Succinate coenzyme Q అనాలి, COQ అనాలి. Succinate coenzyme Q reductase. Okay.
Yes,
Pushpa.
sir.
Complex 3. Pushpa.
Cytochrome C reductase, sir.
Cytochrome C reductase. Next, Baburao. Complex 4.
Cytochrome C oxidase.
Hmm?
ATP synthesis and
Complex 4 is cytochrome C oxidase.
Cytochrome C oxidase.
Oxidase
oxidase,
form.
sir.
Then what is complex 5?
Sir, ATP
Multi-protein.
synthase.
Sir,
Which is, sir?
ATP synthesis. Multi-protein complex.
ATP synthase. So what are the
Synthase.
different mobile carriers?
Synthase.
Different mobile carriers.
5,
Sir,
sir.
NAD, 5, sir, 5 different types, sir. NAD, flavoproteins, iron sulfur proteins, coenzymes Q, and cytochrome, sir.
Very good. How many cytochromes are there? cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3. Okay. So some noise is coming. Please mute
Sir,
your. Okay. So we have discussed about the different components of electron transport system, that is respiratory complexes and mobile carriers. There are 5 types of complexes and there are 5 types of mobile carriers, which are responsible for ultimately the synthesis of ATP. That is the main purpose. What life requires? Life requires energy. What is the source of life or source of life, a source of energy for life? It is the sunlight. This sunlight is converted, that means the light energy is converted during photosynthesis into glucose, and this is called as chemical energy. And this glucose is oxidized, that means in the presence of oxygen it is broken down to release energy plus carbon dioxide plus water. Because oxygen is required here, this is called as aerobic respiration. And this aerobic respiration takes place in 3 steps. One is glycolysis, second one Krebs cycle, and third one electron transport system. Okay, and finally what happens after these 3 things have happened? It is the ATP that is produced. So the immediate, so the source of energy is glucose, but glucose to release energy it requires a lot of time. So the immediate source of energy is ATP, where the adenosine diphosphate, which is having only 2 phosphates, during the breakdown of glucose, during all these reactions, whatever the energy is released, that is used to make a third phosphate bond here, so that the ADP, adenosine diphosphate, becomes ATP, adenosine triphosphate. That means all the energy is present in the third bond. In this third bond, this is a high energy rich bond. So what happens whenever the body requires, it need not go for glycolysis, Krebs cycle, electron transport step by step. It takes a lot of time. So the immediate source is ATP. So it goes for ATP, and ATP is broken down to ADP plus inorganic phosphate. How? By removing this phosphate. As I have said just now, this phosphate is highly rich in energy, and that energy along with ADP and inorganic phosphate energy is also released, which is used by the cell and thereby the body. Okay, and how this takes place? It takes place in the following 3 steps: electron transport, proton translocation, and oxidative phosphorylation. Don't forget, this is the actual sequence. So this is mitochondrion. Protons are released into perimitochondrial space. This is outer mitochondrial membrane. This is inner mitochondrial membrane. This is perimitochondrial space. We have seen that there are 5 complexes, 1, 2, 3, 4, 5 in the inner membrane. And this inner membrane having 5 complexes and 5 different types of cytochromes and other electron carriers, carries only electrons of hydrogen, whereas protons are released into what we call it as perimitochondrial space. So release of protons into what we call perimitochondrial takes place is coupled with transportation of electrons through the electron carriers, isn't it? And all this, what happens here? The ultimate electron transport takes place ultimately to the F0 F1 complex. That is ATP synthase, that is complex 5. And in this complex, what happens? Two events take place. One, the protons present here are translocated inside, which we call it as translocation, proton translocation. They are sent inside. Translocation means sent inside. They are transferred to inside. How? Through the F0 F1 complex. At the same time, along the electron carriers, electrons also come here. So electrons and protons both reach the complex 5, that is ATP synthase. Here, what is electron and proton when they combine to form? They combine to form hydrogen, isn't it? So this hydrogen now combines with oxygen to form water. Okay, it combines with oxygen to form water. And during this movement, what happens? The head of the F0 F1 complex, just a minute. నవ్వించేస్తే అలాగా లైవ్ క్లాస్ యూట్యూబ్ లైవ్ వెళ్ళిపోతుంది. నాకు 10 మంది సబ్స్క్రైబర్స్ ఉన్నారు, రోజు చూస్తారు ఇది. ఆ, అందుకే ఆపేసాను. చెప్పండి. వస్తాను. రీసెర్చ్ వేనా ఫోన్ చేస్తుంది ఆవిడ, లలిత మేడం. ఆహా. నా నా జీవిత చరిత్ర అంతా ఆవిడ దగ్గర ఉంటుంది. మీరు పని మేడం ఆవిడకి చెప్పిన ఒక 20 minutes అయిపోతుంది. ఆన్లైన్ ఇక్కడ నెట్ కదా, రఫ్ చేసుకోనా? Okay, sorry for the interruption. So hydrogen is transferred in the head portion, that is F1. F0 F1 complex is said to be, F1 particle is nothing but a matchstick like structure. It has a base, it has a stalk, and it has a head. And protons are translocated, at the same time electrons are translocated. They reach this place. And by the time they reach this place, what happens? They combine, they couple with each other, electrons and protons, to form into hydrogen. This hydrogen combines with oxygen, and thereby what happens? Water is formed. And during this process, energy is generated. And this energy that is generated is utilized to transfer inorganic phosphate to adenosine diphosphate, so that ATP is synthesized. So what we call this as phosphorylation. Addition of phosphate to ADP is called as phosphorylation. During, during the same time, what is happening? Oxygen is said to be coupled with hydrogen to form water. And hence, as oxygen is required for this entire process, this is called as oxidative phosphorylation. Thus, three things happen here. Electrons are transported through electron carriers of inner mitochondrial membrane. Protons are translocated from the perimitochondrial space into what we call the F0 F1 complex or F1 particle simply. So through electron carriers, through proton translocation, both of them reach the F1 particle. In the F1 particle, both of them combine to form hydrogen. Okay, and this combines with oxygen to produce water. As this is happening, the energy that is produced, how the energy is produced, in the last slide we are going to, I'm going to explain. So the energy that is produced is utilized to phosphorylate ADP by the addition of inorganic phosphate, so the ATP is synthesized. Thus, this ATP is having all the energy that is required for the body. So, let us see step by step how this takes place. So, electron transport system. conversion of one glucose molecule during glycolysis. So glucose molecule to carbon dioxide via glycolysis and citric acid cycle yields a total of 10 NADH and 2 FADH2 molecules. So by the end of glycolysis and Krebs cycle, glycolysis results in pyruvic acid. Pyruvic acid is converted into acetyl coenzyme A. Acetyl coenzyme A enters the mitochondrial matrix, and where it combines with oxaloacetic acid, forms citrate, and the cyclical reactions take place, and again oxaloacetic acid is regenerated. And during this entire process, that means here and here, NADH and FADH2 are produced in both. How many? 10 NADH and 2 FADH molecules are produced. Okay, so what is happening now to these? I have already told you NAD should be available for the next cycle to occur in free form. FAD should be available for the next cycle to occur in free form. But they are said to be engaged. Engaged to what? Hydrogen, isn't it? So NAD has to release hydrogen, FAD has to release hydrogen, and this takes place during electron transport system, and this process is called as oxidation. Oxidation of what? NAD and FAD, where the hydrogens are removed, and these are accepted by coenzyme Q, let us say so. Then what happens? Coenzyme Q is reduced, isn't it? After that, coenzyme Q releases hydrogen and transfers it to complex 3. Now that what happens? Complex 3 gets reduced and coenzyme Q gets oxidized. Thus, there is a sequence of oxidoreductase reaction, oxidation reduction reactions. So the same substance gets oxidized by removing or by releasing hydrogen. Okay, and another substance by accepting the hydrogen gets reduced. So these sequence of reactions go on the entire uh, in the entire electron transport system. So during respiration, what happens? Electrons are released from NADH and FADH2. Why electrons are released? When hydrogen cannot be transported to complex 5 in the hydrogen form, it is split into electrons and protons. And in the form of electrons and protons, it is transported to complex 5, where they combine to form again hydrogen, which combines with oxygen form water ultimately. Okay. So what is happening now? The overall reaction is like this. NADH, H plus H plus, this one releases hydrogen and is converted into NAD plus. That means it undergoes oxidation, isn't it? Likewise, FADH2, this is also releasing hydrogen. This is also releasing hydrogen so that it gets oxidized, isn't it? So oxidation of FAD and NAD plus takes place. And what happens to the hydrogen? This hydrogen combines with oxygen. So we cannot say actually oxygen cannot be half, but we say that during this entire reaction, here hydrogen is released. For water to be produced, only one oxygen molecule is required. And for both of these two together, one oxygen molecule is sufficient. Both of these together combined with one oxygen molecule. And just we have divided it in between these two. But actually there is no half photo. Okay, to make you understandable what is happening here, we have divided the oxygen molecules into two halves, but actually there is only one molecule where these two transfer hydrogen to oxygen so that water is produced. That is the overall reaction. So first of all, it starts with ionization of hydride atom. The mitochondria transfer electrons from NADH and FADH2 through a series of electron carriers. Where they are present? They are present in the inner mitochondrial membrane. Electron transport begins when a hydride atom is removed from NADH and FADH2. This hydride atom then undergoes ionization and splits into proton and electron. This you have to remember. So the hydride atom splits to form proton and electron. This is the first step. So what is the mode of transport of these electrons and protons? While electrons are transferred through series of electron carriers, which we have already discussed, Protons are pumped into the perimitochondrial space. This perimitochondrial space is fluid filled, and this is also called as intermembrane space because it is present between outer and inner mitochondrial membrane. It is also called as intermembrane space. Thus, electrons are transferred through electron carriers, and protons are sent out into the fluid filled intermembrane space, which is also called as perimitochondrial space. So protons are released here, electrons are transported through the inner mitochondrial membrane. So how actually we define electron transport? The stepwise movement of electrons via a series of electron carriers from where from NADH and FADH2, ultimately it releases, they are transferred to what? To oxygen is termed as electron transport. So whether it is proton, whether it is electron, the ultimate receiver is oxygen. So it has to reach oxygen. So here we are referring not to protons, but in electron transport we are referring to only electrons. So what is electron transport? It is a stepwise movement of electrons through electron carriers from where? From NADH and FADH2 to what? Oxygen is called as electron transport. So electron transport is always coupled with proton translocation. So while electrons are transported through the membrane, protons are released into the perimitochondrial space. So electron transport and proton pumping and finally ATP formation all occur simultaneously. During electron transport, proton translocation from matrix to intermembrane space takes place. That is what we have discussed just now. And the intermembrane space is also called as periplasmic side or P side. Periplasmic side or P side. Then where the proton translocation, we have seen that electron transport takes place through electron carriers present in the inner mitochondrial membrane. Then what about the proton, where it gets actually translocated? It is said that it is found that 10 protons are transported out of the mitochondrial matrix. This is the matrix. From the matrix, how many? 10 protons are translocated into the perimitochondrial space. How? For every 2 electrons transferred from NADH to oxygen, this takes place. So from complex 1, 4 protons are transferred. This is complex 1. It transports 4 protons to outside. That means to the fluid filled intermembrane space. And from complex 3, not complex 2, from complex 3, 4 protons are pumped out. And from complex 4, 2 protons are pumped out. Thus, about 10 protons are translocated to the intermembrane space. And this is coupled with oxidative phosphorylation. So, here coenzyme Q and 3 electron complexes 1, 3 and 4 pump protons out of mitochondrial matrix. Complex 1, 3 and 4 participate in the oxidation of NADH and produce 3 ATPs. Okay, when NADH is broken down, when NADH is involved, 3 ATP are produced because there are 3 uh, places of proton translocation. Whereas FADH2 oxidation involves complexes 2, 3 and 4 producing only 2 ATPs per each FADH2. Let us see how this happens. So what happens at complex 1? So complex 1 passes electrons from NADH to coenzyme Q. Complex 1 passes electrons from NADH to coenzyme Q. So this we already have seen here. So from NADH, okay, the electrons are transported to what? Coenzyme Q, complex 1. From the complex 2, they are transported to coenzyme Q. Okay. So the electrons first flow from NADH to FMN, we have discussed it already, then to the iron sulfur cluster, and finally to coenzyme Q. So from NADH to FMN to iron sulfur cluster, finally it reaches coenzyme Q. This sequence we have to remember. So the overall reaction is NADH plus coenzyme Q, and the 2 hydrogen ions that are removed from here, they are transferred to coenzyme Q so that it becomes reduced. Okay, so what is happening? NAD removes or releases hydrogen, it gets oxidized. Coenzyme Q accepts hydrogen and it gets reduced. This is what is happening in the entire event of electron transport system. Oxidation and reduction are simultaneously taking place. And during this place, this time, what is happening? 4 protons, as 2 electrons are transported from complex 1 to coenzyme Q. What is happening? 4 protons are transported into the perimitochondrial space. For each NADH oxidized by the complex 1, which is also called as NADH coenzyme Q reductase complex. The same thing happens in complex 2. Complex 2 is nothing but succinate coenzyme Q reductase. So complex 2 passes the electrons to coenzyme Q. So from complex 2 also, electrons are transported to coenzyme Q. So succinate plus coenzyme Q. What happens? Succinate is converted into fumarate, and during this reaction, what is happening? Coenzyme Q is reduced. So thus, succinate is said to be oxidized and coenzyme Q is said to be reduced. Okay. Next, coming to the complex 3. Complex 3 transfers the electrons from coenzyme Q, which is reduced, to cytochrome B566, then cytochrome B562, 5 iron sulfur cluster, and finally cytochrome C1. Okay, so this is the sequence. So complex 1 sends it to coenzyme Q, complex 2 sends it to coenzyme Q, and from there coenzyme Q transfers the electrons from, so complex 3 transfers electrons from coenzyme Q to cytochrome B first, 566, again cytochrome B, 562, iron sulfur cluster, and finally it reaches cytochrome C1. And finally, from cytochrome C1, these electrons are transferred to cytochrome C, so that the cytochrome is reduced and coenzyme Q is oxidized. And during this process, 4 protons are translocated from the mitochondrial matrix into the intermembrane space. And coming to the complex 4. Complex 4 transfers electrons from cytochrome C to the final acceptor, that is oxygen, and yields water. Complex 4 transfers electrons from cytochrome C to oxygen. Oxygen is the final acceptor, thereby H2O is produced. For each pair of electrons transferred, 4 protons are translocated into the intermembrane space here also. Thus 4 plus 2, that is 10 protons are translocated into the intermembrane space. And finally, what happens? Oxidative phosphorylation. Where it takes place? It takes place in the complex 5. What is complex 5? ATP synthase. It is also called as ATP synthase. What does it mean? It is an enzyme. ACE means enzyme, which is involved in the synthesis of ATP. Hence, it is called as ATP synthase. And this is accomplished by, this is the cristae of inner mitochondrial membrane, which has F1 particle. So throughout its length, the each crista has a number of F1 particles, and F1 particles are the sites of oxidative phosphorylation. It depends on pH to O ratio. So what is pH to O ratio? pH to O ratio refers to the number of inorganic phosphate molecules. That means phosphate which is not bound to any organic compound. It is free, let us say so. So number of inorganic phosphate molecules utilized for ATP generation for every atom of oxygen consumed, isn't it? So electrons are transferred to, along with protons, to what? They are transferred to oxygen. So for every oxygen atom, certain amount of electrons are received, and during this, what is happening? Inorganic phosphate molecules are utilized for the synthesis of ATP from ADP, isn't it? So in it, we can define peace to O ratio not in terms of what we call oxygen consumed. We can define it in terms of electrons transported also. So it is the number of peace to O ratio is the number of molecules of ATP synthesized per pair of electrons carried through electron transport chain. So either by relating inorganic phosphate to oxygen consumed or by relating number of ATP synthesized to the electrons transferred, you can define P2O ratio. P2O ratio can be defined in both the ways. Okay, so P/O ratio is said to be it can be P it can be 3 or 2. For NADH, the P/O ratio is 3. That means when NADH is getting oxidized, it produces 3 ATP. For FADH2, the P/O ratio is 2. That means the number of ATP that are produced is 2 ATP. So if you have 10 NADH produced during Krebs cycle and glycolysis, how many? 30 ATP are produced. Okay. If only 2 FADH2 are produced, how many? Into 2 ATP, 2 into 2, 6 ATP are produced. That means the overall ATP that are produced here is said to be 36 ATP. Okay, and the P ratio of 3 can be summarized as this. NADH releases hydrogen ions and gets oxidized, and this transfers this to oxygen so that water is formed. During this, inorganic phosphate is transferred to adenosine diphosphate. How many? Three are transported. Three inorganic phosphate molecules are transported to, are added to three adenosine diphosphate molecules so that 3 ATP are generated. Thus, the P/O ratio with respect to NADH is said to be 3. And the P/O ratio of FADH2 is 2. That means it can produce only 2 ATP. Why is this so? The terminal oxidation of NADH results in the formation of 3 ATP molecules, whereas oxidation of FADH results in only 2 ATP molecules production. Why? Because it bypasses the first phosphorylation site. Next, what are the sites of oxidative? Let us see what are the different sites so that we can understand how the FADH2 bypasses the first phosphorylation site. So there are three reactions in the electron transport chain that are exergonic. That means exergonic means those that release temperature. Okay, exergonic reactions which result in the formation of three ATP molecules. So when the temperature is released, what is happening? There is release of energy and the work is said to be performed. That is what takes place when temperature is released. That some work is performed and that is utilized in the synthesis of ATP. So there are three sites for ATP formation. One is oxidation of flavin mononucleotide which is reduced FMNH2 by coenzyme Q, and the second one oxidation of cytochrome B by cytochrome C1, and finally cytochrome oxidase reaction. Okay, so these are the three steps. From the substrate, NAD is produced during glycolysis and Krebs cycle. From there it is transferred to flavin mononucleotide, and from there it is transferred to coenzyme Q. So FMNH2 releases hydrogen, transfers them to coenzyme Q, and it gets oxidized. So that we say that oxidation of FMNH2. So oxidation of FMNH2 takes place in the first site. This is the site where ATP is synthesized. And what happens in the next step? Coenzyme Q, which is reduced, now transfers the electrons to cytochrome B and final as cytochrome D, and during which it gets oxidized after transferring the electrons to cytochrome B. This becomes coenzyme Q, and this is the second step where ATP is synthesized. And from cytochrome B, it is transferred to cytochrome C1, C, A, and finally cytochrome A3. And in cytochrome A3, what happens? That is the third site where ATP is produced. So thus there are three sites of ATP synthesis when NADH is involved. And whereas in the case of FADH2, the first phosphorylation site this site is said to be bypassed. Electrons are said to be transported, but protons are not translocated. And that is the reason why here it has a P2O ratio of 2 only, whereas NADH is having P2O ratio of 3. And coming to the mechanism of oxidative phosphorylation, two theories have been put forward. One is chemical coupling hypothesis, the other is called as chemiosmotic hypothesis. And the chemical coupling hypothesis is put forward by a scientist called as Edward Slater. So what this theory actually states, it is still a hypothesis, it is not actually theory. That means it is not having an experimental proof. It is not experimentally proved. So what does Edward Slater say? What did he say? According to Edward Slater, during the course of electron transfer in respiratory chain, he says that a series of phosphorylated, that means those substances to which phosphate is added. Phosphorylated high energy intermediates are first produced, which are utilized for the synthesis of ATP. But actually, it lacks any experimental evidence because no scientist is able to isolate any of the high energy intermediates that was discovered by, that was stated by or hypothesized by Edward Slater. That is the reason why this theory is said to be not in vogue. And the most important theory is chemiosmotic hypothesis. Okay, so this is not all, this is still called as hypothesis, not formed into what we call uh, theory. Why? Because it is still experimental evidences are being going on to support it, but to some extent, when compared with chemical coupling hypothesis, this explains how the ATP is generated. So it is the widely accepted mechanism of oxidative phosphorylation. Who proposed it? It was proposed by Peter Mitchell in 1961. What does it explain? It explains how the transport of electrons through the respiratory chain is effectively utilized to produce ATP from ADP and inorganic phosphate. Okay. So, according to him, the first reaction that is required, the first event that is required is proton gradient. So, this is the mitochondria. This is the matrix. This is the perimitochondrial space or intermembrane space. What is happening? In step 1, step 2, step 3, 4 plus 4 plus 2, 10 protons are transported to outside. We call it as translocation. So what is happening now? Proton concentration increases in the perimitochondrial space. So when the concentration increases on one side and decreases on the other side, there is a difference in concentration. And this difference is called as gradient. Thus, a proton gradient is established. That means here there are more protons, here there are very less protons. A proton gradient is established. Okay. The inner mitochondrial membrane, throughout its length, where the F1 particles are absent, wherever the F1 particles are absent, the entire inner mitochondrial membrane is impermeable to protons and also hydroxyl ions. That is the reason why protons accumulate in the perimitochondrial space. They cannot come, though proton gradient is established. Then what is the way? The only way is the complex 5, that is the F1 particles. So in the inner mitochondrial membrane is folded to form cristae, and each crista is having a number of F1 particles. And these are the places where protons, based on their concentration difference, that is proton gradient, can come inside, which we call it as proton translocation. So the transport of electrons through electron transport is coupled with translocation of protons across the inner mitochondrial membrane from the matrix to the intermembrane space based on proton gradient. How it takes place? The pumping of protons results in the electrochemical or proton gradient. That means more protons are outside the inner membrane and less inside the inner membrane. That means matrix. This is due to the accumulation of more positive protons on the outer side of the inner mitochondrial membrane than the inner side. So the proton gradient developed due to the electron flow in the respiratory chain is sufficient to result in the synthesis of ATP from ADP and PI according to Peter Mitchell. So this is the proton gradient coupled with electron transport is required. What is required for the synthesis of ATP? So what is the enzyme system? How the ATP is synthesized during electron transport coupled with proton translocation. So ATP synthase, which is nothing but the complex 5, utilizes the proton gradient for the synthesis of ATP. This enzyme is also called as ATPase. Yes, it can also hydrolyze ATP to ADP and PA. So this enzyme is having, it can form, synthesize ATP from ADP and PI. It also can break down ATP to ADP and PI. So that means this complex is both synthase as well as ATPase. Let us discuss only, restrict our class only to ATP synthase form. The ATP synthase is a complex enzyme. It consists of two functional subunits, namely F1 and F0. So F0 is the stalk followed by base, followed by a stalk and a head region. This is the F1 particle. The protons that accumulate on the intermembrane space, so in the intermembrane space these are present. They re-enter the mitochondrial matrix, leading to the synthesis of ATP. How they re-enter the mitochondrial matrix? Through the F0 F1 complex. Then what about the remaining inner mitochondrial membrane? It is impermeable to protons. Hence, they can enter the mitochondrial matrix only through the complex 5, that is F0 F1 complex, also called as F1 particle, also called as Fernandez-Molan particle, also called as elementary particle. It is also called as oxisome. So through oxisomes or F1 particles, the protons are translocated into the mitochondrial matrix. Okay, so the protons from the F0 F1 complex, they are transported through this. And what does this actually F1 particle consist of? This is called as P side. This is the inner mitochondrial membrane. This is matrix. This is perimitochondrial space. So this membrane has two faces, two sides. One facing matrix called as M side, M for matrix. The other facing perimitochondrial space is called as P side, P for perimitochondrial space. So there are two sides, M side and P side. And P side is having protons, more protons here, less protons here. That means proton gradient is established. More proton concentration is more. Now what happens? This membrane is impermeable to protons. So wherever the general inner mitochondrial membrane is present, protons cannot come inside. Whereas wherever the F1 particles are present, the protons can be translocated. And this F1 particle in its head region is having 1, 2, 3 alpha subunits, 1, 2, 3 beta subunits, and 1 delta or gamma subunit, one, one delta subunit, one gamma subunit, and one epsilon subunit. Thus, 3 alpha, 3 beta, 1 delta, 1 gamma, 1 epsilon. Thus, there are 9 subunits in this, what we call F1 particle head. So what happens is, scientists say that they have proposed, Peter Michel have proposed, Peter Michel proposed chemiosmotic hypothesis. What is chemiosmotic? Osmotic means where there is concentration of solute is more on one side and less on the other side. What happens then? Water is transport, osmosis takes place. Here also there is concentration of protons on one side. Okay, hence this is called as chemiosmotic. And to explain this, how ATP, this chemiosmosis or proton translocation is involved in the phosphorylation of ADP to produce ATP, Paul Boyer in 1964 proposed the rotatory model for ATP generation. Simply speaking, what actually this is, not going to the intricacies. Let us see cycle. We have a cycle. So the cycle has tires. So this tire is connected to another tire, isn't it? So this cycle, towards its front side, it has a light. So when you cycle, when you go for cycling, what happens? the light flashes. How the light flashes? Because it flashes only when the tire rotates. Only when the tire rotates. So how it takes place? The scientists who invented light for the cycle has placed a small structure called as dynamo. It is placed here. So this dynamo is is said to be placed over the wheel. So as the wheel is rotating, what is happening? Mechanical energy is produced. Mechanical energy is produced. We know that according to thermodynamic principles, energy can neither be created nor be destroyed, but can be transformed from one form to the another form. So here, the mechanical energy that is produced here by the help of the dynamo is converted into electrical energy, and thus that electrical energy is transferred to the light through a wiring system. So the rotatory motion creates what we call it as energy. Likewise, when you deal with what we call hydropower stations, in hydropower stations there are large turbines. So large turbines of water, these turbines rotate water. As the water is rotated in the turbines, energy is generated, and that energy is said to be converted to electrical energy, and we are using that. That is what actually this rotation, this rotatory power has more power to generate energy. That is what is happening here. This is the head portion. This is the crista of the inner mitochondrial membrane. And what is happening? Protons are translocated into the head region. As protons are translocated in the head region, through the inner membrane, electrons are transferred. So electrons reach and also protons reach, where the head portion of the F1 particle. So this F1 particle, according to scientists, is the smallest in the entire universe. It is the smallest rotatory unit. Smallest rotatory unit. Even the Earth rotates around its own axis. Likewise, this rotatory unit, that head portion, rotates on itself. It rotates on itself. This rotation creates certain amount of energy. How the rotation takes place? Because electron transfer and proton translocation, as they came at once, it is both are said to be coupled. When they reach the head portion, it gets rotated, and that rotational energy that is produced is converted into chemical energy to form a bond between, that means between the adenosine diphosphate and an inorganic phosphate to synthesize ATP. That is what the rotatory model is. So according to Paul Boyer, a conformational change. So rotation means what is change? There is a change in the shape. Change in the shape is already called, we have already discussed that. Change in the shape is called as conformational change. Remember, this is O, not I. Conformation, it's not conformation. Conformational change, change in the shape. So, a conformational change in the mitochondrial membrane proteins leads to the synthesis of ATP according to Paul Boyer. Okay. The F0F1 complex, that is complex 5, is also called as ATP synthase because it is involved in synthesis of ATP. The F0 subcomplex is composed of channel protein C subunits to which F1 ATP synthase is attached. So this is channel protein C subunit present in the F0 to which the F1 ATP synthase unit is attached. Thus, hence we call it as F0 F1 complex. The F1 ATP synthase, this one, F1 ATP synthase consists of a central gamma subunit surrounded by alternating alpha and beta subunits. So which we have already seen. This is the stop. This is the stop. A central gamma subunit to which the other subunits are alternately arranged. In response to the proton translocation, we call it as proton flux. Transfer of protons or translocation of protons from perimitochondrial space into the mitochondrial matrix is called as proton flux. In response to the proton flux, the gamma subunit physically rotates. So this pink colored one rotates. It rotates physically. Okay. This induces conformational changes in the beta subunits that finally lead to the release of ATP. So rotation of gamma subunit brings about a change in the beta 3 subunit, and by a mechanism still not known, the energy, that rotational energy that is produced, is involved in the synthesis of ATP and its release thereby. According to the binding change mechanism, the three beta subunits of F1 ATP synthase adopt different conformations. So each beta subunit is having different conformation. One subunit has open conformation, the second has loose conformation, O conformation and L conformation, whereas the third one has a tight conformation. So these three beta subunits have different conformation, different orientations or shapes, we can say. One is open, O conformation, the other is loose, L conformation, the other is tight, T conformation. So by an unknown mechanism is not clearly understood. That is the reason why this is still chemical chemiosmotic hypothesis only, not it is chemiosmotic theory. So by an unknown mechanism, protons induce the rotation of gamma subunit, which induces the conformation changes in all these three conformations of beta subunits. The substrates ADP and inorganic phosphate bind to beta subunit in L conformation. So what happens? ADP and PI are attached to the beta subunit in L conformation. Now what happens? The L side changes to T conformation. L side changes to T conformation. That means this place L conformation, let us say this is having O, this is having L, this is having T. As rotation takes place, as change takes place in the shape, L is converted into T. Okay. The L side changes to T conformation, and this leads to the synthesis of ATP, because already to L conformation site ADP and PA are added, and ADP, anorganic phosphate, has to be added to the adenosine diphosphate, and this takes place when L side changes to T conformation. The O side changes to L conformation, which binds to the ADP and PI. The T side changes to O conformation, releases ATP. So thus these all the three subunits of beta subunit change their change their conformation, that means shape, and release in the release of ATP according to the rotary model. So this cycle of conformation changes of beta subunits is repeated. As a result, ATP are generated for each revolution. For each rotation or revolution, the energy that is produced is utilized for the synthesis of ATP. It is estimated by scientists that for every 5 protons translocated out, 4 are used to synthesize one ATP molecule, and 1 is used to power the export of ATP from the mitochondria in exchange for ADP and inorganic phosphate. Thus, 5 protons are required for the synthesis of ATP according to this model. It may be noted that the ATP release in O conformation is energy dependent and very crucial in rotatory motor for ATP generation. So for here in O conformation, ATP release is said to be energy dependent. That means it requires energy. That means ATP has to be broken down. We have said that the same ATP synthase also acts as ATPase. It has two activities. So thus here in O conformation, the energy required for this conformational change is said to be brought about by the breakdown of ATP. That means some amount of ATP is utilized during the production of ATP event. The enzyme ATP synthase acts as a proton driving motor and is an example of rotary catalysis. That ATP synthase is considered as the world's, as I have said before, it is the world's smallest molecular motor involved in the synthesis of ATP. So this is what actually happens. Somebody please mute your mic. Somebody please mute your mic. So here, during a translocation of protons and transfer of electrons from NAD to ultimately oxygen, from FAD to oxygen, what is happening? Brahma. మ్యూట్ చేయమంటే జాను ఎవరు? You're not listening to the class. You're somewhere else. Then why should you join? Think about this. So there are four complexes and this is complex 5. Protons are released in each step to outside. So proton gradient is established. Okay. And electrons are transferred. Electrons are transferred through the electron carriers. So electrons reach the complex 5, protons also reach the complex 5, and the protons and this entire membrane is impermeable to protons. That means protons cannot be transported anywhere, so they have to be transported only through the F0 F1 complex. So here the protons are coming and electrons are coming, both of which combine to form the hydrogen molecule. And here it combines with oxygen to form water. And during this what happens? The F1 particle, the gamma subunit followed by the beta subunits undergo conformational change. Rotational energy is said to be produced. And this acts as a molecular motor. The F1 particle acts as the molecular motor, the head portion. And the energy that is produced during this reaction is involved in the synthesis of ATP from ADP and inorganic phosphate. This takes place towards the M side. And translocation of protons takes place from the P side, perimitochondrial space. And this entire diagram you have to remember. So I'll share the PPT. You go through the entire topic once, and whatever doubt you have, please ask me in the group. I will answer you, and then we complete the lesson. Any doubts, please ask me. Otherwise, we'll close for now. Thank you.